Welcome to Inert Chemistry. In today's video, we are discussing about the pyrimidine nucleotides. Here first we see a pyrimidine ring. This is a basic structure of pyrimidine ring. It is a single ring structure and having carbon and nitrogen present in it. It is a six-membered ring. Out of these, the three carbon and one nitrogen, these nitrogen 1, carbon 6, carbon 5 and carbon 4, these are contributed by the aspartate, while this carbon 2 it is contributed from carbon dioxide, and this nitrogen 3 it comes from, or it is contributed from, the amino group of glutamine. Remember that unlike the purines, the pyrimidine ring is synthesized as free pyrimidine and later on it is incorporated into nucleotides. Pyrimidine bases. The three common examples of pyrimidines are cytosine, uracil and thiamine. Let's discuss the pyrimidine biosynthesis. This pyrimidine biosynthesis is quite easier than purines. During the first step, carbon dioxide plus glutamine, they combine with each other and form carbamoyl phosphate. In this reaction, glutamine is the amino group transferred. It transferred NH2 amino group to carbon dioxide and itself converted to glutamate. Here two ATP molecules are also utilized and they are converted to ADP, and the compound that is formed is carbamyl phosphate. Here you see the structure of carbamyl phosphate. The reaction is catalyzed by an important enzyme, carbamyl phosphate synthetase 2. It is activated by ATP and PRPP while inhibited by UTP. Remember that there is also an enzyme that is carbamyl phosphate synthetase 1. This carbamyl phosphate synthetase 1, it is used in the urea cycle and it involves in the first step of urea cycle. It is a mitochondrial enzyme, means present in mitochondria, while this CPS2 is present in cytosol. Here are the key differences between CPS1 and CPS2. In the second step, the carbamyl phosphate that is formed, it then fused with aspartate. By the help of enzyme aspartate transcarbamylase, and carbamyl aspartate is formed. Here you see this whole blue structure is actually aspartate that incorporates into carbamyl phosphate and form carbamyl aspartate. In the third step, water molecule is released and ring closure occurs. By the help of enzyme dihydroorotase, and the compound that is formed is called dihydroororate. Remember that these three enzymes, carbamyl phosphate synthetase 2, aspartate transcarbamylase and dihydroorotase, these are the three domains of a single enzyme. So it is a very important example of a multifunctional enzyme. In the step 4, this dihydroorate is converted to orotate. In this reaction, NAD plus is used and it is converted to NADH. The enzyme that is utilized is dihydroorotate dehydrogenase. In the fifth step, this orotate is converted to orotidine monophosphate. In this reaction, PRPP is utilized. Remember that this PRPP is formed during the first step of the purine biosynthesis from ribose 5-phosphate. Ribose 5-phosphate is reacted with ATP and PRPP is formed. So in this fifth step, this PRPP is utilized. This PRPP donates the ribose 5-phosphate to orotate and the resulting compound that is formed is orotidine monophosphate. It is also called OMP. In the sixth step, carbon dioxide molecule is released. Here you see COH is present, but here is CH, means one carbon dioxide molecule is released here from here, and OMP decarbox by the help of enzyme OMP decarboxylase, and the resulting compound is uridine monophosphate, are also called UMP. These orotate phosphoribosyl transferase and OMP decarboxylate, these are the two domains of single enzyme. So it is a bifunctional enzyme that having two domains. Any defect in this bifunctional enzyme causes a disease that is called orotic aciduria, in which orotate level is high in the blood. In the reaction 7, 8 and 9, this uridine monophosphate is in the reaction 7, this uridine monophosphate by the help of enzyme kinase is converted to uridine diphosphate. Here phosphate group donor is ATP. In the 8th step, it is converted to UTP, and similarly then it is converted to CTP. When UTP is converted to CTP, glutamine is used. Glutamine donates the amino group to UTP and it is converted to CTP. Cytidine triphosphate. Similarly, this UDP is converted to deoxy UDP. This reaction requires thioredoxin, and this deoxy UDP is further converted to deoxy UMP, and then it is converted to deoxy TMP by the help of enzyme thymidylate synthetase. In this reaction, N5N10 methylene tetrahydrofolate is used. Thanks for watching this video. In the next video, we will see about the regulation.